Hi, and welcome to this special edition of the Moving Science Podcast. I am Lior Freeman, an MS specialist and researcher based in Austin, Texas, and I am privileged to serve as your host today. This is my first time in this role, so I beg for your kindness in advance. And today we're here to highlight one of the 5 projects that have been selected for funding by the MS Innovation Challenge. I have here 3 brilliant minds and inspiring humans to discuss the innovation they are moving forward together in MS. Dr. Christy Sheehy, Dr. Jacqueline Thies, and Dr. Ari Green are exploring how retinal eye movement can help us detect MS progression earlier. Their project is called a quantitative oculomotor biomarker of PIRA in individuals with MS on B-cell depletion therapy. Today, we're going to unpack the problem they're trying to solve and how their solution, the RetiTrack device by Sealight, has the potential to transform MS care. Dr. Sheehy, Dr. Thies, and Dr. Green, welcome. So I'd first kind of like to go around and have each of you tell us a little bit about yourself and of your background, and we'll dive a little bit deeper into all of this in a minute. But Dr. Sheehy, kind of in a few words, tell us about you.
Hi, everyone. I am Christy Sheehy. I'm the CEO and co-founder of a startup called Sealight Technologies. My background actually started off in optical engineering. And so the core technology that we're going to be talking to you today all about is the RetiTrack. And I actually created that during my PhD at UC Berkeley. After finishing my PhD there, I then went on to do a postdoc in neurology at UCSF and really start to understand the amazing connection between the eye and the brain and focusing in on ocular motor health for multiple sclerosis. And I'm joined here by my amazing colleagues, Dr. Thies and Dr. Green. Dr. Thies is our Chief Medical Officer, so I'll pass the baton over to her.
Hi, I'm Jacqueline Thies. I am a residency-trained neurooptometrist, and I first started working with Christy at UC Berkeley, where we did research with her original device, looking at eye movement fixation stability in elite-level athletes. And since then, I have become the Chief Medical Officer for Sealight Technologies and also work clinically. And I have worked clinically working with patients who have functional impairments from ocular motor dysfunction, particularly from neurologic or developmental abnormalities. And I'll pass the baton to you, Dr. Green.
Thanks, guys. So I'm Ari Green. I'm a neurologist and physician scientist at the University of California, San Francisco, where in addition to overseeing our research program in multiple sclerosis, as well as our clinical and educational programs in MS and neuroimmunology, I work in my own laboratory in trying to develop and validate biomarkers for monitoring in MS. So our primary goal is to develop tools that are capable of helping us to monitor disease and prognosticate related to disease and even potentially diagnose disease better. So principally, we're just really want to help with the advancement of new therapeutics, but we recognize that in order to get new therapeutics, we need to be able to measure response to therapy and need to be able to measure disease more precisely.
Awesome. Well, thank you all. As I mentioned earlier, we're going to talk a lot about retinal eye movement as a potential biomarker for MS. And I have to admit, I'm an MS specialist, but I don't know very much about retinal eye movement. So I'm going to start a little bit on the MS side of things, because that's easier for me to understand. And I'm going to ask you, Dr. Green, you talked about your lab, wanting to advance how we treat people with MS, by developing biomarkers. Your proposal is centered around MS progression. And I want to ask you, why tackle MS progression? What is it? And why is it a problem worth solving right now?
Yeah, so MS is unique, I think, amongst diseases, neurological diseases with a progressive phenotype or significant progression and degeneration and worsening, in that we've developed therapies that are helpful in treating the diseases. But principally, those diseases focus on the immunological factors that drive attacks in the disease, but they aren't all that successful at, or they aren't successful enough in preventing progression. And that remains our kind of major unmet need in the field, which is how do we best help people stop progression from happening and keep them from having worsening disability, especially as they age. So you see that as one of the major, maybe the major unmet need in the field. And as a consequence, we really need tools that are capable of measuring that progression. And the main problem is if you wait for the progression to happen, you might be trying to intervene too late. So we need precise, more accurate, and very sensitive tools and techniques. And that is really where this technology comes into play.
I know that's really interesting and really relates to our practice. I mean, we know from the literature, but also from our day to day that, it may take years for us to even recognize that progression is occurring. And it would be so much better to be able to identify it early. So Dr. Thies, you are a neuro-optometrist. And that sounds fascinating. And I feel like you have a lot to teach me and teach our listeners about eye movement in general and retinal eye movements in particular. And I'd like to first understand why studying eye movement matters kind of more generally. What do eye movements tell us?
While people think about the eyes and they always just see them at the front of the face, people don't realize just how much of the brain is dedicated to moving our eyes and interacting with what we see. And so the visual pathway within the brain expands the entire neural network. And depending on how you look at eye movements, whether I move my eyes from the left to the right, if I move them quickly, if I'm tracking something while I'm looking, I'll use different parts of the brain. And so the neat thing about using the ocular motor system is that it actually has a pretty good potential to be an indirect biomarker of neurologic function, especially if you can use technology to look at the subtleties like the velocity and the speed and the direction and the accuracy. All of those little nuances of the eye movement are actually controlled by the brain. And so when we think about how we interact with the world, we have that afferent pathway. So bringing the signal into the brain through the afferent visual system. And then the brain decides how it wants to respond and sends that efferent signal back out. And it uses every lobe of the brain, brainstem, cerebellum, midbrain. And so it's a really neat diffuse network. And then when we look as far as function goes, everything we do in life, for most of us, we're using our sight to do it. And I think one of the big things that is under-evaluated in patients with MS is their visual function. So clinically, I work with a lot of patients who have subtle eye movement problems, whether it's from MS, Parkinson's, concussion. And a lot of the symptoms that they end up getting because their eye movements are inefficient with visual instability would be unsteadiness with walking, hesitancy with steps, not feeling safe while they're doing things, or just feeling slow to read, slow to interpret. And so there's a lot of function that goes on when we have abnormal eye movements that I think we're clinically missing. And it's not going to show up on an MRI, and it's not going to show up on some of our standard tests. And where I think the potential for having eye tracking comes in, and we'll talk a little bit about retinal eye movements, which I think people don't know much about. But the big potential for me is going to be in quantifying some of those problems. Because a lot of times, by the time a neurologist sees the nystagmus that the patient has from their MS, and they send it to an eye care provider, right, an ophthalmologist or an optometrist, by the time that your visual system can see it in another visual system, it's a pretty large movement. And that movement is enough to cause oscillopsia, or the feeling that the world is moving around them. It can make images blurry. No matter what prescription I give them, I can't make it clear because the blur is coming from motion. And so there's things that we can try to do functionally to help the patient, but at that point it's too late, kind of to Ari's point of trying to treat the disease before it has progressed so far. So I think there's a lot of potential of if we can see it sooner and on a more microscopic level, then we can intervene sooner. So I think that's a big part.
It's fascinating. Basically, you're telling me that what I'm doing in clinic, just having people track my finger, really is not helping anyone.
I think it's helping. I think we just haven't had the technology available, honestly, to be able to give any better data than what we already have. I think that's been the big barrier with a lot of other eye trackers, is that a lot of times those eye trackers are using large structures to track. And so the information they were giving us wasn't anything different than what our clinical exam was already giving us. And so what would be the utility of those if me having you follow my finger is the same equivalent to that? I do still think there's value in the clinical exam for sure. Where it comes really is that prognostication. Because for me, it's, if I see nystagmus and I do a treatment for the patient, the only biomarker I have to see if my treatment worked is if the patient says they feel better. I can't be like, oh yeah, that slowed it down. Like I can't detect speed necessarily. I can say slow or fast in my clinical exam. Maybe Dr. Green's better at it than I am, but I think some of those nuances are hard to use if you're trying to see if your treatment is actually working.
Yeah. And to your point, if we're looking at prediction, as Dr. Green highlighted, we need quantifiable measures. We need, and that kind of takes us kind of to the technology that you guys bring to the table. So I want to talk about the technology, but I also want to talk about the innovator behind the technology. And that kind of brings me to you, Dr. Sheehy, because, I think that you, as you highlighted earlier, you've invested kind of a lot of time in your career to studying this, to trying to bring this product, the RediTrack, to life. I'm going to ask you about the solution itself, but please first tell me a little bit about your story, because from what I have heard is kind of a story of passion and grit. And I think that it can inspire a lot of our listeners today. So tell us a little bit about how that came about and your own personal story as a founder.
Absolutely. So, with the technology in grad school, I had created we call it a tracking scanning laser ophthalmoscope, a TSLO. And it was a way to track eye movement without having to use any of the fancy kind of high resolution adaptive optics that we were currently using in our lab. And so many times to see the back of the eye, to see photoreceptors, you need to be able to use adaptive optics. And so my project was basically the only one in the lab. Show me how you can get high resolution retinal imaging without having to use that. So that was kind of the core premise of what I was doing, is how can I make it cheaper, more compact, more clinically viable? And so for me, that was a really exciting starting point, because it allowed me to think of ways in which I could truly translate it to clinic. And so during my PhD, I would present at conferences. I would talk with different, professors, different clinicians. And many of them at the time had me go to their site and build one of our devices. So my postdoc at UCSF with Ari, I built a device at UCSF there. I had built a device in Hong Kong. I had gone to Germany to build the device. And after a little while, I started to realize, hey, if people want me to build this for them, maybe this could actually be a viable business. And so I started doing some of the accelerator programs that they had at Berkeley, which is a fantastic ecosystem for entrepreneurs. And it really has that entrepreneurial spirit in the Bay Area. And talked with my PI at the time, Austin Rorta, and said, hey, I'm thinking about spinning this out into a startup. Would you be supportive of this? And he was absolutely hands down supportive. He said, I'll be here, to back you up in whatever way you need, but I'm not interested in business, so go do your thing. And so kind of the wheels were off and running starting in grad school. And the first way I knew how to get funding was through grants. So I applied to NIH. I did a lot of the NIH small business grants to start, just to really get an idea of what I was thinking for eye motion and how it could be impacted by neuro would work. It was a way in which I could have non-dilutive funding and kind of test that hypothesis, and had an STTR with Ari here to look at how fixational eye motion changed during the course of the MS disease. And then shortly after validating and proving that out, we actually became venture-backed in 2021. So we then had venture capital invest in C-Light. And from there, it's really been a roller coaster in terms of how to take a technology truly from bench to bedside, how to take it from the lab and make it into an FDA-cleared product, how to make sure that you're creating something that a clinician wants to use. That's why we have Jackie's expertise, on the team as well, making sure that what we're providing the clinician makes sense for their workflow, their day-to-day use. And so for me as a founder, starting off in science, it was an interesting ride. I think I first thought everyone would be as interested in the technology as me. But you quickly find out when you're pitching to venture capital, it's really about the business. And so that kind of shift in my mindset in terms of how can I, in one or two slides, convey the premise of this amazing technology, but also show the potential it has for clinic, the potential it has to make revenue, to be billed through insurance and do other, revenue generating activities, really became the driver for how we could create the business. So it's definitely been a roller coaster. And I think a lot of PhD students if you're passionate about your project and you're worried, is my thesis just going to collect dust? It's definitely an opportunity to spin out and start with NIH grants. And if you're able to prove something using non-dilutive funding, you're kind of off to the races at that point.
Yeah, no, this is so inspiring for those of us who've just been in academia our whole life. It's it's a complete different world just to hear about, just trying to make sense for the clinicians and trying to make sense for investors and all of that. So thank you so much for sharing. I think that there are a lot of our listeners that may be wondering if their invention might be a good business venture. And I think, they would love to, they appreciate you sharing your story. But so tell me more now about, I haven't really seen the Reddit track. I mean, I've seen it, but I haven't really played with one, so it kind of looked like this quite convenient kind of tabletop device. So tell me a little bit more about the device itself and what are retinal eye movements? What does it measure? Exactly. That would be great to understand.
Absolutely. The instrument itself is tabletop, as you mentioned. It looks a lot like the types of instruments you would see at an eye doctor. And so the beauty of our device is that it's completely non-invasive. There's no eye drops, no puff of air. People always ask me, "You gonna blow air in my eye?" No, we're not. It's completely non-invasive. You just put your chin onto a chin rest, and then you stare as stable as you can at a target. And so we record 10-second videos. Each of them is done monocularly, so one eye at a time. And then from those high-resolution retinal videos, we're locking on to individual cone photoreceptors. And so those are the structures that we're using as a reference to track. And so if you think about traditional methods using the pupil, you could almost think of it as tracking your hand versus tracking your pinky nail. If I have a smaller, more robust target, I'm going to have less fluff or less noise around that tracking signal. And so by locking on to individual cone photoreceptors and capillary networks, we're able to see eye motion all the way down to 0.1 degrees of visual angle, which translates to about 30 microns or less than half the size of a human hair. And so it's amazing to me, even at that level, you can have individuals complaining of the world jittering, oscillopsia, the world shimmering is something I've heard as well. Even that small of a change, if it is something new or acquired that is happening during the course of the disease, patients can experience it. And there's no clinical tool right now that can really show objectively what that movement deficit is until this technology has come to light. And so by using the retina, it's a fantastic way to look at subclinical, very subtle eye movement abnormalities at the fixational level and provide that objectively to the clinician, not only in terms of metrics like amplitude or speed, but also for patterns. So as Jackie had mentioned, looking at nystagmus, having a waveform where you have the drift and snapback kind of in a very repetitive fashion, or potentially a square wave jerk train where the eye is kind of just bouncing back and forth. And so being able to provide those objective metrics and the pattern to the clinician allows them for a really streamlined ocular motor output that they just have never been able to see before. And to me, that's the most exciting part, is bringing new healthcare data into the space. We're not, creating something that's objective that you already know or you already do. We're bringing you new data that can help drive different and novel decisions.
Yeah, that's great. And I can see how, the fact that it's relatively simple to use and it doesn't have a high burden on patients or clinicians can make it fit into a clinic workflow relatively easily. So Dr. Green, you were with Dr. Shi from the very beginning of the development of this device and this technology. Sounds like there was a lot to figure out at the time and how to make sense of this information. So tell me, what drove you to work with Dr. Sheehy? What happened in those early days as you guys were trying to figure it all out?
Yeah. Well, I can tell you, I remember exceptionally well when Christy, this young engineer, came to speak to me at a conference in Florida. And what struck me about her from the outset was her enthusiasm and her engineering technical wizardry. Those things were obvious, and she could talk about things that were exceptionally exciting. And she made us think about how could this tool actually be useful in clinical context. But then we started to collect data, and the thing that was really, I think, daunting to us was the amount of data that came out of the machine. And that's where my perception of Christy as an enthusiastic and capable engineer changed into recognizing her as a trailblazer, because she was thinking about, how do you take huge amounts of data and turn it into something that's actually useful in the clinical context? How do you make it digestible? And she was way ahead of the curve thinking about big data. This is, going back almost 10 years, 2016, 2017. She was thinking about, how do you take big, daunting amounts of data and turn it into something that's analyzable, and thinking about what we would probably now call an AI approach. But at the time, we were thinking of just as let's take the tons of data that comes out of this thing and make it into something useful. And she really was just way ahead of the curve on that front.
Yeah, incredible, very visionary. And, Dr. Thies, AI is this hot topic right now in healthcare. I'd love to understand more from your perspective, like, what is the role that you think AI plays in eye tracking, and how does that make your innovation different?
So one of the things that I've loved about Christy's intuition on the future of eye tracking is that she's recognized the need for machine learning and AI technology from the very beginning. And so I think the first thing is just getting the technology out there. But then, as Ari mentioned, one of the things is that you get so much information. You get amplitude, size, speed. We're taking multiple scans, or we have multiple eyes. And I think one of the things that we recognize in healthcare that we never want to tell our patients is that we can't see all things or recognize all patterns. And I think the real future in AI and eye tracking is recognizing what we don't see and being able to weed out some of our clinical biases. And the key that I think Christy's done a great job at is we've been doing research on AI since 2017, looking at how we can use AI a little differently with our data. And she's hired some of the best people in AI and technology development within our company. And I've learned a lot from them and understanding how AI works, because I'm a clinician. I'm not an engineer or a software guru by trade. And I think what's been interesting is learning the concept of garbage in versus garbage out, and how you have to be very selective about how you train these models and create these models. And that's where we've been working really hard to make sure that we work with the top level clinicians when we're working in different disease states doing research, so that we know that we're getting good data in to feed our algorithms, and making sure that in the end we're creating something that's useful but also valid and precise. I think right now everyone's saying, oh, we have AI. And I think learning with this company, I've learned you have to kind of ask, well, what does that even mean? And how good is your AI? And just like I would say you would criticize the method section of any paper that's done on a study, you should do the same type of criticism when someone's quantifying that they have AI, and ask, how does it work? What did you feed it with? And those, I think, are the new things that we as clinicians don't know we need to ask about AI algorithms when we're bringing tools into healthcare.
I mentioned earlier, your team was selected for funding by the MS Innovations Challenge. So, first, congratulations are in order. This is huge. This is really awesome. So my understanding is that this funding will help support a clinical validation study in MS and progression, as we highlighted earlier. So, Dr. Green, can you tell me more about the study? What is the hypothesis? What are key aspects of your design? How are you going to go about it?
Yeah, so we've come to understand with highly effective immunomodulatory or immunosuppressive therapies that we can stop relapses from happening. We can almost entirely eliminate them. But the problem that persists, as we touched on earlier, is that patients continue to progress. And so the field as a whole has come up with a new conceptual understanding and a new insight into the fact that patients throughout the course of their disease, in fact, have progressive, insidious worsening that occurs. And we've named that PIRA. So this concept of PIRA, or progression independent of relapse activity, remains this way in which we can monitor and at least define who's getting worse. But the thing that we're lacking is, how do we know if PIRA is going to happen in the future? how do we have an indication that someone is heading towards progression that is going to be obvious enough for the physician to notice at the bedside in a brief examination? And so the idea is to use this highly, highly sensitive tool, this technique, in a population of patients who have early disease. So either have had one attack or have had maybe an attack plus some other features that prove that they actually have MS. So early MS patients, and identify whether or not the eye tracking tool, the TSLO, is able to, is it associated with the development of PIRA, and is it predictive of PIRA over the course of a few short years? So that's really the design and intention of the experiment, and then looking at how it tracks and relates to other measures that we already use for monitoring early disease and that worsening and progression that still happens despite use of highly active therapies. So these patients are all going to be on very, very active therapies to suppress the big scale worsening of relapses. But now we're going to see, can we track the small scale worsening that is manifested in PIRA?
Yeah. So I saw in your proposal that you're not only going to try to predict progression based on our classic kind of ambulation related scores like the EDSS, but you're also looking at how, those retinal eye movements can help predict cognitive decline, for example, trying to relate it to cognition. Why is cognition important in MS?
Yeah. So there's a number of areas in MS where we don't track all that well. We don't monitor all that well. As you touched on, the EDSS is great at monitoring motor worsening and motor progression and walking, worsening in walking or ambulation. But it's not so good at tracking the declines in function like with cognition. And it's really because the declines in cognition in MS are much slower and much less obvious than they would be in diseases like Alzheimer's disease or other classic neurodegenerative diseases of the elderly. So in this regard, we need tools that are going to be useful for tracking the slow cognitive decline that occurs. And it's really important to note the types of eye movements that Christie's technology allows us to track. These fixational eye movements, their underlying biological basis is much less well understood than it is for other types of eye movement abnormalities. So maybe put really simply, we tend to think of eye movements as one thing as neurologists, as frontline clinicians, but it really is it's a number of different functions that are all kind of lumped together as eye movements. So those include, how do you move your eyes in order to pay attention to a new thing that's interesting? That's a saccade. How do you track an object that's interesting? That's a pursuit. But then there's also, how do you stay with stable fixation, stable maintenance of your eye position on something that you're paying attention to? Well, you see with my introduction of the word attention there's almost a planning and a cognitive component to what's required for maintaining fixation. And so it's a long way of saying, in order for us to track fixation, we are probably tracking things that are related to planning, attention, maintenance of the interest. And those are all potentially subtler features that are useful for monitoring cognition. So we need better ways to monitor cognition, and there are ways in which we can detect features that are probably relating to people's cognitive dysfunction in MS that we can monitor with a tool like TSLO.
Very interesting. I think that's going to really resonate with people who live with MS and who live with these kind of planning attention difficulties, and sometimes they're fine otherwise, but this is what prevents them from living the life that they want to live, or even staying employed, and so that really matters, I think. And it's part of this invisible disability that's associated with MS, and I think that's really 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 interesting. um so Dr. Thies, kind of walk me through what happens next. So clinical kind of validation studies often kind of the first step towards care transformation. If your study is successful, what's next? What needs to happen for your solution to have an impact directly in the lives of people living with MS?
So, as you mentioned, I think the big thing for us always is we need to establish the clinical validation and the biomarker credibility. I think for other eye tracking issues, there's always been issues with repeatability and precision. And I know we don't have that as many issues with that because we don't use the dynamic structure like a pupil or a corneal surface like other eye trackers do. So I think we have more promise in that validity. And to Ari's point, I think one of the big things that we will have to do next is also a lot of disease state education in fixational eye movements. I think even eye doctors don't realize that just looking at something, you are actually making a very complicated cognitive motion. So I always call fixational eye movements the kind of the Goldilocks of eye movements. If you always need the eye to move, otherwise you'll neurally adapt to the visual signal. But if the brain makes you move too much, right, you get nystagmus and oscillopsia. But if you don't move enough, you'll get visual fading. So it has to constantly monitor how much motion it's doing. And it is actually relative to the task and relative to a lot of other complicated things within our system. And I think what's exciting first is going to be understanding if it's a great biomarker. It's one of the most objective biomarkers that we can do because you're just telling someone to look. There's no voluntary kind of plateau, which we sometimes have with other ones where getting someone to do something if they're cognitively impaired, they just can't do the task and therefore you can't track that biomarker. So I'm excited for this one. But I think we really need to work on disease state education for people understanding. I think people hear retinal eye tracking and they think that we're essentially an OCT tracking the retina. And it's not that at all. We're using the retinal structures to track microscopic eye movements, and then getting people to understand that we're making these movements all of the time and they're neurally regulated. The next step, usually for most companies, would be kind of FDA, at least in the United States, FDA regulatory clinical pathway integration. Luckily, we've already hit our head against that wall so far and have been really successful. So we've gotten our device cleared as an FDA eye motion monitor. And then we've also done regulatory work for a CPT and reimbursement code. So we did go to the AMA. And we're able to get our own code that will launch in 2026 because we are a novel biomarker. There's no other biomarker like it on the landscape. So I think a lot of it is going to be those clinical steps. And there's a lot of hurdles we've found, I think, because eye movements span so many different disciplines, right? They're going to present in neurology, they're going to present in ophthalmology and optometry. And it's trying to get the interest, but also getting people to understand that the technology can help your clinical assessment. And I think for us, it's always been, and this is my own clinical kind of experience, I'm not going to bring a tool into my office unless it helps a patient or it changes my decision making, in which case then I'm going to buy it. And I think the big thing for us is we just need to validate that this tool can do that for you and then kind of get the clinicians to see it. But I love that this is something that can be used in research and something a clinician can use, because I get frustrated when you have a lot of these trials that come out and the endpoints that they're using or the devices they're using to track their endpoints are not clinically available. And then as a clinician, it's then hard to be like, okay, well, they were able to show that the drug worked, but how am I going to be able to do it? Because that tool only exists in that one academic center. So I'm excited to be able to work in this study and make sure that we create something that can be usable in research and bedside.
Yeah, I love this kind of thinking about the implementation, the clinical, just not just a validity, but kind of the implementation, the clinical workflows, and that broader utilization. What needs to happen to make that a reality? So thank you for clarifying already kind of all the work that you've already done and are currently doing and thinking, and how you're going to kind of broaden access. I want to kind of close up on kind of expanding a little bit our horizon about this tool and retinal eye movements. And, I know Dr. Sheehy, as a founder, as an innovator, I'm sure your vision for this solution goes beyond MS. And you know, as Dr. Thesis highlighted, you know, there's going to be other specialists interested. There's probably going to be other subspecialties within urology where this is kind of applicable, and this kind of power of scale probably gives more, you know, just more of a motor towards implementation than we think about as, you know, people like myself as academic in my very narrow field. So tell us a little bit more about your work outside of MS and why it matters as well.
So the beauty with eye motion is that the literature has shown for decades that there are changes with many different diseases. So scientifically, we already know that there's saccadic and smooth pursuit changes in neurodegenerative disease, psychiatric disease, looking at, you know, individuals with amblyopia, strabismus, retinal disease. So there's this beautiful culmination really in the literature showing just how powerful eye motion can be for a host of conditions. And so for us, the tricky part was, you know, where do we start? When you have something that can change and be impacted in so many different conditions, how do you pick kind of that first beachhead market? And we did choose MS because it was an area where there are viable therapeutics out there. So if we find changes, if we see things that are happening and we inform a clinician, there's actually something you can do about it. And so in the neurodegenerative space, it was a no-brainer for us really to focus in on MS first because of that therapeutic landscape that already exists. But as we're starting to see, we're getting more drugs now for Alzheimer's. We're starting to get the amyloid drugs. And I think over the next 5 to 10 years, we're really going to start to see a shift where my hope is that neurology goes from kind of more reactionary, where people that are already sick are in the waiting room, to more proactive and more preventative. How we can start to see changes earlier on. And that's really going to help drive the drug development landscape, which in turn then impacts the stage of the patient that you're going to see in clinic. And so, you know, my big vision for the future, pun intended, is that every year you would go to get your physical, you check your physical health, you check your heart health. No one really checks your brain health. And so is there a non-invasive way in which we can truly assess cognitively how we're functioning and have it be something where each year you can longitudinally track that? The best way in which you can keep track of cognitive health is comparing it to yourself. And so I think while having a normative database is going to be something that's fantastic that will continue to grow, longitudinally tracking yourself over time is really going to be that true north for precision medicine. And for me, that passion really comes from having a family with neurodegenerative disease. So I've had a grandmother pass from Alzheimer's. I've had a great aunt pass from Alzheimer's. I had my aunt have early onset Alzheimer's at only the age of 60 and pass away. And now one of my immediate family members actually has MCI. And so for me, being able to bring a tool to the neurodegenerative stage to try to really focus in on how can we help patients? How can we use a tool that can see very subtle change to drive therapeutic intervention to be streamlined and give you the best solution as quick as possible to limit disability? I mean, for me, that's the big win. And so driving from that personal passion point and being able to remember that day to day in our work is really what keeps me going. And I think there's going to be a lot of excitement as we expand to different conditions in the future. And the research is already there for larger eye movements. We're just going to bring in these smaller eye movements and show their validity as well.
Yeah, awesome. No, thank you so much. I cannot imagine a better way to close this conversation. Like to think about, brain health in general and the health of our population and how, eye tracking may be this window into the health of our brains. And so thank you all so very much for this enlightening conversation. And I wish you the best of luck on this project. And thank you to a listener for tuning in today.